So our next speaker is Howard Sirotkin from Stony Brook University.
So in mammals, the maternal environment has long-lasting impacts on gene expression. Factors including maternal stress, nutrition, insults like smoking affect the chromatin landscape of the developing embryo. There are also genetically encoded factors that impact the epigenome of the embryo. We've been interested in the role of early transcription factors that are maternally deposited in control of gene expression in the embryo. In particular, one factor called Mrest or NRSF. Mrest is a repressor protein that binds to an approximately 23 nucleotide DNA element termed an RE-1 site. And in most vertebrates, there are about 1,000 RE-1 sites, most of which are associated with neural-specific transcripts. Mrest recruits two repressor complexes mediated by SYN3 and CoRest that include a number of chromatin-modifying factors, including HDAX1 and 2, G9A, and as well as a number of other chromatin-modifying factors. When Mrest was first identified by the Mendel and Anderson labs, it was thought to have a main role in repressing expression of neural-specific transcripts in non-neural tissues. In addition, the Mendel lab and others have subsequently shown that Mrest regulates gene expression in neural stem cells and progenitor cells, delaying expression until differentiation occurs. And lastly, Mrest-mediated repression can be regulated based on neuronal activity. So several years ago, we used a now antiquated technique called zinc finger nucleases to make a Mrest mutant. And the mutants we made produce a truncation very early on in the protein. We're unable to detect protein by immuno, and we think they're likely to encode a null allele. And we noticed that Mrest mRNA is maternally deposited, and we're very curious as to why this neural-specific transcription factor would be maternally deposited and be potentially active very early during embryogenesis. And so for today, I want to talk about our efforts to try to understand the role of this maternally supplied Mrest in terms of its influence on larval and adult behavior, gene expression, and then talk about our efforts to identify key targets of Mrest that modulate behavior. So the fish that I'm going to be talking about most often today are these fish in here that we refer to as MZrest. So these are the product of a Mrest homozygous female times a wild-type male. So they lack maternal Mrest, but are zygotically hets. And so we feel the proper control for these animals is the reciprocal cross that we refer to as ZRest hets. So this is the product of a Mrest homozygous male times a wild-type female to produce animals that have wild-type levels of maternal Mrest, but then are then heterozygous for zygotic Mrest. And then we also occasionally look at these maternal zygotic Mrest mutants. So these are the product of two homozygotes and lack both maternal and zygotic Mrest. When we look at the behavior of Mrest larvae at 6 days, so just looking at spontaneous movements of 6-day larvae in the light, and we do this in a 24-well format. What we've noticed is that these animals are hyperactive. So shown here in red are the Mrest, comparing them to the ZRest animals. This is just looking at the number of total movements. And we see that the Mrest make about 1,400 movements over 15 minutes compared to the ZRest, which would make about 1,000 movements in this interval. This is simply a breakdown, minute by minute, binning of this data over the 15 minutes of the assay, and we see that at all time points, the Mrest fish initiate more movements. If we also looked at other parameters of motion, time swimming, distance traveled, it would look just like this, and that the Mrest are more active. In addition, we've noticed a spatial preference for the Mrest larvae, that they tend to prefer the periphery of the wells. So here's some representative path diagrams of 3 ZRest larvae and 3 Mrest, and where the well is just artificially divided into an inner and an outer region. What you can see is that the ZRest larvae have a greater tendency to explore the center of the tank, as an event that's seen less frequently in the Mrest. So this is at 6 days, but what was really surprising to us is that we would still, in these Mrest animals, see behavioral defects as adults. So now we're looking at locomotor path diagrams of 6-month-old animals and in a novel environment assay. So we're looking at the movement of a single fish. This is 5 minutes of movement. And we noticed something really interesting. So if we look at the female ZRest or female Mrest, we see very similar kinds of movement patterns. However, the Mrest males compared to ZRest males behave very differently. So the first thing you can notice from these movement diagrams is that there's a position bias, and that the Mrest adult males tend to prefer the periphery of the tank. Once again, it's not shown here, but they also swim very erratically, so they tend to have rapid vertical swimming motion and tight turning angles and several other parameters of erratic swimming. Again, if we look over the course of the 15-minute assay in 1-minute bins, comparing the ZRest males to the Mrest, sorry, this is females, ZRest and Mrest, we don't really see a difference, but there's a very clear difference between the Mrest and ZRest adults. So there's something that maternal rest does very early on that has lifelong impacts on behavior. So we're very interested in trying to understand what the genes are that might be responsible for these behavioral phenotypes that we observe in Mrest. So we did an RNAseq experiment, and the logic of the experiment was we did this at sphere stage, so shortly after the midblastular transition, so that we felt that by doing it that way, most of the targets we would find were likely to be direct effects of rest and not secondary effects. And then we looked at 4 different sets of animals. So we compared Mrest, the transcriptome in Mrest, to ZRest, looking for things altered in Mrest. And then we also compared the MZrest mutants to wild type, looking for things altered in MZrest. And then we looked for the overlap between the Mrest and the MZrest as sort of a secondary check, the idea that most transcripts that are misregulated in Mrest should also be regulated in MZrest. And we identified a set of 158 transcripts that are upregulated in both Mrest and MZrest, shown here. And of those, we significantly enriched for RE-1 sites, which is our expectation that most bona fide rest targets should have RE-1 sites. We found 63 genes that contained RE-1 sites. We looked at these by qPCR at 14 of them. 13 of them we were able to detect signal for, and all 13 of those were misexpressed in the same way by qPCR. So 63 is still a pretty sizable set of genes. So we established some additional criteria. First, we looked at genes that were involved in behavior, and in particular, we noticed that many of these genes were found at synapses. I'll talk about that more in a second. And then we also looked by qPCR with the assumption that genes of interest would be misexpressed beyond sphere stage. So we focused in on a couple of these transcripts, actually 3 of them, that are misregulated, or transcripts that are genes that are involved in the synapse. So 2 of them are snare components, Snap25a and Snap25b. And the third one is amplifizin, which is involved in vesicle recycling. What we see is that all 3 of these genes, as I said, are misregulated at sphere stage, and this misregulation persisted. This is looking by qPCR, where the Mrest is set to 1, and then doing a relative comparison to the ZRest. And we see for all 3 of these that misexpression is still detected at 8 somites. However, in the case of Snap25a and b, once at these early stages, there's very low levels of expression, and what we're really seeing is precocious expression here. Once transcription really gets fired up, we don't see the same overt changes by qPCR, at least. However, in the case of amplifizin at 6 days, we still can detect by qPCR upregulation of amplifizin in Mrest fish. If we look by RNA in situ hybridization, so now looking at Snap25a and b, this is a dorsal view of the hindbrain of Mrest. at 24 hours. And we see a sort of subtle phenotype, but it's very reproducible, that we see these slight increases in SNAP25a expression, also SNAP25b shown here compared to ZRest. We don't see changes in amplifysin at 24 hours, but if we do the same in situ at 6 days, where we saw the difference by qPCR, we see a fairly robust difference by in situ hybridization. So obviously there's a lot of genes that, and a large set of genes that are regulated by Mrest. And we wanted to find a way to specifically block the REST interactions with specific target genes. And so to do this, we utilized the CRISPR-Cas9 system. So the idea is that REST binds to the RE1 site and induces the laying of repressive marks that then repress gene transcription. So we reasoned that if we deleted out RE1 sites, we would leave all the other regulatory elements that govern expression of these particular transcripts intact. We'd leave the coding sequence intact, but they would be impervious to REST. And so we did this for both SNAP25a and SNAP25b. In the case of SNAP25a, we generated fish that have a 12 nucleotide deletion that takes out over half the RE1 site, including a number of residues that are essential for REST binding. For SNAP25b, we isolated a larger deletion that takes out the entire RE1 site. When we looked at the effects of removing the RE1 sites on expression of these genes, we saw expression patterns that were very reminiscent of what we see in the Mrest animals. So here's looking by qPCR early on. It's fear stage, and we see that SNAP25a without the RE1 site is upregulated, as is SNAP25b. We see a similar effect. And then again, looking at 24 hours by RNA in situ hybridization, we see this very subtle but consistent expansion of the expression domains more immediately in the case of SNAP25a and SNAP25b. We wanted to know whether there were the same kinds of effects on behavior. And so this is the same spontaneous movement locomotor assay looking at animals that are homozygote for either the SNAP25a or the SNAP25b RE1 site mutation. In both cases, so here's the wild type, and here's the mutant siblings for SNAP25a and b. And again, we see very reminiscent kinds of hyperactivity to what we see in the Mrest animals. The overexpression of SNAP25a or b was shown by the Patton lab at Vanderbilt to produce hyperactivity in very much the same way. And in addition, they showed that conditions that led to overexpression of SNAP25a or b produced hyperactivity and alterations to the primary motor neuron architecture. So we wanted to look at the primary motor neuron architecture in Mrest and as well as in the RE1 site mutants. So this is a lateral view of the trunk at 56 hours. We see as an increased number of punctus. So these are neuronal increased numbers of neurites on the motor neurons in Mrest. And we see a very similar, although a little bit weaker effect in SNAP25a, suggesting that this phenotype may be responsible for the locomotor enhancement that we see in the Mrest, that SNAP25a is the key target. We don't see the same effect in SNAP25b, suggesting that some other aspect of SNAP25b regulation is responsible for this, for the phenotypes that we see. So in conclusion, in maternal REST is expressed early as a maternal transcript, and then later it's a zygotic transcript. But there's something special about what happens really early on with REST that zygotic REST is unable to compensate for. So we think one of the differences here is that prior to these genes being transcriptionally active, REST is able to influence these loci in ways that it can't later on. And this produces effects in both gene expression and behavior. So I'd like to lastly acknowledge the people who did this work. The majority of these experiments were done by a very talented graduate student, Kara Morvik, who just graduated and is moving on. The adult behavioral studies were assisted by our collaborators in Ingenious Targeting Laboratory, Wei Wang. The bioinformatics, we had help from Ian Woods at Leeds and John Samuel at Seneca College. Thank you.
Thanks. Sorry, that was very elegant.
Thanks, Rob.
What's your model? How does it work that you turn on the zygotic expression just a little while later and it's completely unable to do its thing? So your something about early programming.
Yeah, exactly. So this is obviously where we'd like to go next with this, and we'd like to understand. So I think that the thing that changes, in that period is that transcriptional activators come on, and so we think that when the chromatin is more compact, perhaps Mrest is able to reinforce or add marks that are going to be long-lasting. And so then you have these activators come on, turn things on, and Mrest can no longer has that same access to the chromatin. It's still bound, probably. We haven't verified that, and we want to do that. But maybe even though it's there, it can't do quite the same things that it can once there's the presence of other activators.
Thank you. We'll have to move on.